It's so noisy. Great, that's now the first thing in my broadcast. But whatever, I stand by it. Was it really? Actually? I'm having a rough go for making myself look bad in broadcast form. All right. So we're going to get into the idea of genetics. And remember, genetics is how we pass on what molecule carries the information, our blueprints? They code to make proteins, but it is the DNA or the RNA that actually carries the information. And when we say pass something on, what do we mean? What's that mean, pass something on? Sorry? Yeah, for sure. That's what we're talking about. Yeah. Yes, say it again. Pass it down from whom to whom? Yeah. Like from dad or from mom, and from them from grandma or grandpa or great-grandma or great-grandma. That's right. Down your family line. And if you remember, one of our central beliefs in biology is cell theory. We talked about this a bit when we talked about what makes something alive. But remember, cell theory basically says, A, that all living things are made of cells. Another thing is that cells are able to reproduce. Very good. Remember, that was one of our things that we said. And so this leads to a conclusion. If everything's made of cells and cells are able to reproduce, then all of the cells and things you find, all of your cells, all of my cells, those bacterial cells in there, every cell that we see, where did they come from? They came from older cells reproducing, just like people, right? Every person is the product of reproduction, right? If you meet a person, they have parents, and their parents had, and they had, and they had, right? So all cells come from, or are the product of, is another way of saying, previous generations of cells reproducing. So when we say that each cell is the product of a previous cell reproducing, that means that every cell's DNA, which we remind ourselves is the blueprint, comes from its ancestors. That should not be an apostrophe. Sorry, while I was talking, I just looked in here and saw how absolutely hideously filthy the bottom of this was. And I thought I'd take it upon myself to clean it so it would stop bugging me. That's all. That didn't go well at all. Okay. Now, eventually, in our next unit, we're going to get to the complicated case. And what are you guys?
Complicated. Nobody understands me.
All right? I don't want to talk about you yet. The way that animals reproduce is a lot more complicated than what we're going to talk about today. The way that even plants reproduce is a lot more complicated than the way we're going to talk about today. Today we're going to review what you probably learned in grade 9, which is asexual reproduction. So at its most basic, here's the idea. We have to have new cells must form with a complete set of DNA. Which is all the same DNA as what we call the parent cell. Well, if you're going to make a new cell with new DNA, that means that DNA must be reproduced, must be replicated. As, I'm sorry. And there should be a the while we're at it. So the DNA has to be replicated first. We're not going to get into that in great detail, though can I quickly say how it works? In asexual reproduction, a perfect copy of the parent cell's DNA is made. We don't have to get into this too much, but remind me, DNA, how many different letters are there? How many different bases can a DNA molecule be made from? 4. Adenine, guanine, cytosine, thymine, A, G, C, and T. So, and how many strands are there? DNA is always found with the bases, not singly and alone, but there's 4 different ones. But if you remember, if I make a row, like say I had DNA and this is A, G, C, G, C, C, A, T, and like this is just a section of it. Could you tell me what's going to go here? What would this be? Do you remember? They pair up. They always are 2 strands, and A is always with T, always together. That means G must always be with. So that's a T. This would be a. This would be a. This would be. That's a G. Yeah, this is a C. This is. Cool. So when the cell wants to reproduce, all it has to do actually is kind of split this. And if you make it so that it splits up, now this is complicated because your DNA is like meters long inside a tiny cell, but all the same, if you split these apart, what can now happen to each of these strands? So we split the DNA open, we untwirl it and we split it open, and now new DNA can get written in here so that what we end up with is the original strand here, and then its original pair here, and each of those will have had partners put on it. So this A would now be with a, this G would be with a, this C would be with a G, C, G, G, T, A. And here we'd have A, G, C, G, C, C, A, T. And so the DNA splits, and new bases added, forming a perfect copy. But interestingly, each perfect copy is actually half old DNA, half new DNA. Does that make sense? If you look here, this is half original and half new, and this is half original and half new. That's how it's done. This isn't super important, but does that make sense? That happens to all of your DNA. Now here I've shown it with what, like 9 letters? Your actual DNA, hundreds of millions of letters. So it's messy, it's complicated, but this is the first thing that happens. A copy of the DNA gets made. Does that make sense? And in asexual reproduction, it is a perfect copy, which means that in asexual reproduction, the offspring is going to have the exact same DNA as the parent. Do you have the exact same DNA as your parent? If you did, what would you look like? You'd be a clone. You would be exactly them. Does that make sense? It'd be like you were your parent's identical twin, which would be weird, right? You'd be like them exactly, but like 18 to 37 years younger kind of thing. Be weird, right? That's not how sexual reproduction works. When we get to sexual reproduction, we're going to see that you're a combination of some DNA from your biological mother and some DNA from your biological father. But in asexual reproduction, all that happens is inside the cell, all of your DNA splits apart, and because bases always pair up in a certain way, we can take the half of the DNA molecule and just build a new second half to it, and now this is a perfect copy of the original DNA. We do the same to the other half, and now it is also a perfect copy of the original DNA, and you take one copy of your DNA and turn it into 2. By the way, machines have to come along and do this. What are those machines made out of? What are the doing parts of cells? Proteins. What are proteins made out of? The individual building blocks are amino acids. How did the cell know how to put the amino acids together to make that protein in the first place? Where did it find that information? Sorry? Yeah, which are the DNA. So somewhere in this DNA, there's a gene that says, hey, put this amino acid, now put this amino acid, now put this amino acid. And when you read that section of DNA, it builds a machine whose job is to come in and split DNA into 2 pieces. And then somewhere else on your DNA, there's a section where it says, read here, that's an amino acid, that's an amino acid, that's an amino acid. You do that 800,000 times, and you get another machine whose job is to come in and where there's a single-stranded piece of DNA like this, add all the bases to turn it back into a complete piece. And the same over here. Those are called DNA helicase, whose job is to break it apart, and DNA polymerase, whose job is to add new bases. You don't have to know that, but it's kind of cool that like the machines that do this were built using the instructions in the DNA. Excuse me. All right. Once the DNA has been replicated, now the cell can break apart. Does that make sense? If the cell breaks apart before we have a second copy of the DNA, is that going to work? Can you make 2 cells? No. So this has to happen first. So, once the DNA has been replicated, the cell can start the process of splitting. And of course, it's a big title on this page. What do we call the process by which a cell splits into? Mitosis. Very good. By the way, we call the original cell the parent cell. Here's some gendered language. We call the offspring the daughter cells. This is the process of mitosis. We're going to talk about eukaryotic cells. Bacteria are kind of boring. They have no nucleus. Their process is way simpler. Bacteria split by a basic process called binary fission. They basically copy the DNA, split in half, and they're done. Does that make sense? This is simple. Don't draw this. But for a bacterium, their DNA just sits loosely in the nucleus, and it tends to form a loop called a plasmid. And all it does is it makes a copy of that loop. So it's like, oh, now we've got one copy of the loop and a second copy of the loop. And then it just splits in half. Oh, now we have this cell with one loop and that cell with another. You don't have to draw this, but see how easy that is? Very boring, right? Called binary fission. Binary meaning two, fission meaning being cut in two. But for eukaryotic cells, remind me, what's that term mean? Eukaryotic cells are prokaryotes are the simple ones. Eukaryotes are the complicated. You don't have to write this. They have nuclei and other organelles. So these are cells with a nucleus and other organelles. And if you're going to cut that cell in half, you're going to have to deal with the nucleus, right? Would it do any good to double the DNA, cut the cell in half, and one cell gets the nucleus with two copies of the DNA and the other cell gets no nucleus? Is that going to work well? No. So we are going to talk about that. So let me ask you this. Anyone ever moved from one house to another, one apartment to another? Yeah, me too. A few times when I was a kid and a few times as an adult. I don't know about you, but I have a lot of crap. Anyone else have a lot of stuff? Stuff in your drawers, stuff in your shelves, stuff in your closet. You guys handle your closets the way my kids handle their closets, which is by taking everything they own and just shoving it in there and then shutting the door and pretending it's not a problem. And if you say, hey, can you clean up your room? They go, sure. And they go like, rah! And they just shove everything in there and like kick it and then shut the door. No, you don't do that? Do you do that? No? Anyone here do that?
I used
my
to.
kids. Yeah, when you were like 7, right? Yeah, my kids don't do that. Anyway, but to be honest, so do I kind of. I don't do it in my closet, but I have a drawer. They don't have like a drawer on the desk where whenever they don't know where something desk goes, it goes in there and it's just a big pile of chaotic mess. Anyone? Ah, yeah, I like that. So listen, if you're going to move house, are you just going to start shoving all that stuff in the car and drive it across to the other house? Or what do you do? You're going to move. What do? You take all your stuff and you into boxes, right? You put a bunch of stuff, put in a box, and you take it. Right? Does that make sense? And then even then, all your boxes, you're probably going to put in one pile, right? So they don't get mixed up with your stupid brother's stuff and you put all his stuff in another pile, right? I don't know if your brother's stupid, but he probably is. I don't have a brother, but if I did, my sister has a brother, and that guy's stupid. Right? Okay. Anyway, you put all your stuff in boxes, and then not only that, but you separate the boxes so that like everyone's stuff is sorted and ready to go, right? Why do you put it in boxes? Why bother? You're just going to take it out of boxes. Doesn't it seem wasteful? Why do you put stuff in boxes? Just to take it out of boxes when you get where you're going. Right? So things don't get broken, and so things don't get lost, or things don't end up in the garbage or the wrong place. Does that make sense? Well, when the cell is about to divide, it's about to move house. Does that make sense? And the first step the cell wants to do is to pack up the most important stuff and make sure it stays safe, right? If you're moving, I bet you spend a lot of time packing the stuff you really care about, right? Things that are important to you. And then the stuff you don't really care about, maybe on the last possible day, you just like shove into a box and you're like, yeah, whatever. Does that sound accurate? That's certainly what I do, right? Like my guitars and like my things I care about, like my photo albums and stuff like that, I do like a good careful job packing. And like my clothes and like crap like that, just like the day before, it's like, oh shoot, just throw it in a box, right? What's the most important thing to a cell? The single most important thing in it, if it's going to survive and be able to make the machines it needs and do all its work, and if it's going to be able to reproduce. The single most important thing is the... What do you think? Say it. God, Matthew. What do you think? Yeah, and specifically what inside the nucleus? The DNA. In fact, the reason we have a nucleus is because when the cell's living its normal life, we want to keep the DNA safe because the DNA is so important. It's the blueprint of how to make a cell. If it breaks, the cell can't work. But when we're going to move, we're going to have to take it out of the nucleus. And before we take it out of the nucleus, we want to pack it up. Does that make sense? So, before the cell splits, the DNA is coiled tightly. and supercoiled. Oh man, what's that mean? To make it compact and to protect it. What's it mean to make something compact? That's right. So to give you an example, if we had DNA looking like this, and obviously it's two-stranded, that's DNA, we coil it so that it goes like this. Picture like a telephone coil. This original one would have been much longer. And then we take that and we coil it. So this is coiled, and this is supercoiled. Do you know when I say a telephone cable, do you know what I mean by that? Because, of course, your phones don't have cables. But you know what I'm talking about? Like the one that like spirally like this? Why did they used to make telephones with cables like that? With the wires that connect the handset to the base? Why do you think? More cable, less space. Does that make sense? It was coiled like this, so it didn't take up much space. But when you lift it up, you could walk far away and it would stretch out. And then you're done, you could bring it back and it would like spring back. Because that, like, you used to have to do that, right? Like the phone would be in the kitchen on the wall. If you want to talk to somebody, you'd have to take it off the wall. But maybe you also want to like make dinner, so you'd have to like walk around with it. And there's a big stupid cable there. So you could buy like long cables. They'd be coiled like this. Now imagine you took a telephone coil and you coiled that again. Does that make sense? That's called a supercoil. And in fact, it's done even one more time, which I'm not even going to attempt to draw. I'm just going to say again. And what you end up with is, instead of all the DNA in the cell, which just looks like this, a big chaotic kind of mess, gets coiled into chromosomes. So we call this the chromatin. That's loose DNA. And we call this the chromosome, which is tightly packed. And then remember, we said that the first thing that happens before the cell can split is we take the DNA and we make a copy of it, right? So actually the chromosomes don't end up looking like this. They end up looking like this because it is copied to a copy of itself. We could say like copy 1, and this would be copy 2 because it's been replicated. Does that make sense? And now, some cells only have one chromosome. All the DNA fits on one of these. Do you know how many humans have? You're really close. 23 is right. So, the number of chromosomes differs from cell type to cell type. Now, you're going to see me put a number up here, and it's going to seem like it contradicts what Matthew just told me, but actually it agrees with what he just said. Humans, for example, have 46 chromosomes. But Matthew said 23, and both are kind of true answers. You see, we have this weird thing where, yes, we have all our DNA separated into chromosomes, but you know that you don't get any of your traits directly just from mom or directly just from biological dad, because we actually have 2 copies of 23 unique chromosomes. One from mom, one from dad. What word did I just put there? Yep, which means what?
From the mother.
Yep, and the other?
Paternal from the father.
Very good. From mater, which is Latin for mother, and pater, which is Latin for father. Go home and you could say, hello, mater. Hello, pater. They'd probably be like, why the hell are you talking like that? Stop. My daughter's mad at me. She refers to me as father. So if I've annoyed her, she'll be like, father. I'm like, shut up, Claire. But I don't know if she does that to my wife or not. I've never actually paid attention to whether she gets mother. Probably. Sorry? Like you will or you do? Do you? You go like mother. And does she or does she go like? There you go.
I do that sometimes if she doesn't even blink. Like, I called her spawn point multiple times.
I like that. Spawn point.
At one point, that was the only thing she could remember. Like I would yell, "Mom, mom, mom, mother."
Which means I just want to show kind of the progression here really quickly. We're going to skip all the steps and we'll fill them in later. So we start with a simple cell and it's got loose chromatin in it. It is going to take all that loose chromatin and turn it into chromosomes. Now we actually have 46 of these. Oh, sorry, I didn't mean to do that. We actually have 46 of these, but I'm going to show 2 because I don't want to draw 46 stupid little chromosomes. Does that make sense? And then we said before the cell can split, we have to take that DNA and we have to make a copy of it. What will these look like now? Well, they actually stay stuck to their original copy. So in the DNA copies, you get these stereotypical chromosome shapes that are X-shaped. Does that make sense? And that's one copy stuck to a second copy. And now here, we're going to go through the process of mitosis. And then after mitosis, when all is said and done, what we're going to have is 2 cells that just have one copy each. And then they're going to want to do their job. So they're just going to chill out and go back to just loosely packed chromatin mode and live their happy little cell lives. Does that make sense? Until they're ready to reproduce. Does that kind of make sense? So if we wanted to put a couple of notes on here, it's not some big deal, but here the DNA packs to chromosomes. And again, you don't have to write this necessarily. I'm just making sure if you look at the note later, you'll know what it means. This is where the DNA makes a copy of itself. And then mitosis is the process by which they split. And then, of course, what can both of these cells do when they're done living their happy little cell lives and they're ready to reproduce? They can each pack their DNA into chromosomes, make a copy of itself, undergo mitosis, and these 2 cells could eventually turn into 4 cells, right? 2 each. And those 4 cells could eventually turn into, and those 8 cells could eventually turn into, and 16 into, and 32 into, 64 into, 128 into, 256 into, 512, 512 into, 1024, 1024 into, 2048, 2048 into, 4096, 4096 into, 9128. No, I screwed up. Okay, we'll move on. Anyway, it doesn't matter. Point being, I'm bad at math.
Well, you were speedy at it.
Right? So, we'll just quickly get into this. All of this, if we imagine this as a cell's life cycle, all of this is called interphase, and interphase I do not actually want to draw it like that. I want to show it. But interphase is the boring part of a cell's life where it just does its thing, right? So, yeah, I just want to remember how I spaced this out below. Oh, I left myself lots of space. So this really is just the diagram. Okay. Cells live their normal cell lives in what we call interphase, which tells you a lot about how we think of cells. Because it's weird. It's almost like how we think of people, right? Where like once you have kids, you're no longer like a real person. You're just like somebody's dad or somebody's mom, right? Like it's like you stop having your own life. You're like, oh, that's Claire and Greg's dad. And you're like, I'm a person. I have a name. Especially as a teacher, like you don't have a first name anymore. You're Orlando. And like not like here, but like if you saw me at the mall, you could be like, hello, Joseph. And I'd be like, why, hello. Why, hello, Ms. Colombe. Yeah, I don't know why we can't just be called by our first name, but it's considered like unprofessional. I don't really get it. I don't see why it was like with the school collapse into chaos if you just called me Joe and you got like, you know what I mean? But it's just not how we do it. So what do I know? I don't know anything, right? But you think about it, it's kind of weird, right?
I think it's like a respect.
Like everyone gets it. Yeah, I guess. When I taught at South Lincoln, they always called me Sir. That was like a real country school thing. Yeah, I used to too, and now I'm just used to it. But yeah, at South Lincoln, it was always, excuse me, sir. And I was like, sir, I like this. Never been called sir at Eastdale. But anyway, maybe once or twice. Okay. But it's only at the very end of interphase, or I shouldn't say at, but near the end of interphase, that the DNA is replicated. We call this the S phase of interphase. And S is for synthesis. I don't really care about that. Can I tell you? I don't really care about this. After the DNA replicates, if a cell's going to split in two, before it can do that, it's got to get a bit bigger. Does that make sense? So after the DNA replicates, the cell grows larger in preparation for splitting. And just like people, these things can last different amounts of time. Some cells live happy little cell lives for a little bit and then very quickly replicate their DNA, get bigger, and then split. Bacteria do that. You're seeing that back there, right? Your bacteria are growing really quickly. Other cells will spend years in this G1 phase, which is just doing their regular old cell activity. Does that make sense? Your brain cells, most of your brain cells are as old as you are. You know, especially the ones in your core root ganglion. They just spend a long time here. They don't normally get old and reproduce. In fact, your brain cells, most of the ones you have now, will be with you when you die. They just kind of stay with you. Your body does replace some as they get damaged, but a lot of them will live for tens and dozens of years. Does that make sense? Other cells get to the reproduction stage very quickly. I think about this. I went to Eden where we had a lot of like Dutch Reform kids. They all seem to be like married and having kids by like 18. And my wife and I were married and we had kids by like 23, which is still pretty young. And then like some of my friends didn't have kids until they were like in their late 30s, right? Just different people spend different amounts of time in each stage, and cells are no different. Does that make sense? So different cells move through these stages at different rates. Bless you indeed. Each and every one. No, I don't think so. I hope not. Yeah. Sure, yeah, of course. No, but I mean, like, and you can replace them or whatever. But yeah, no, like brain cells can be damaged like any other cell, right? And if they're damaged, then they either can be replaced, ideally, or sometimes aren't, which is problematic. Anyway, using this as an opportunity to beat up on your sister in a class she's not even in. Regardless, the actual splitting, well, we don't have to write that. Let's just get into it. The actual splitting is called mitosis. Okay, we're going to go through the phases, and you probably learned this before, but I just want to go through it, and I want to make sure, because if you try to memorize this crap, it's hard to remember, right? If you're like memorizing it. Whereas, if you tell a story, it can just make sense, you know? If you think about mitosis as what has to happen, it just makes sense. So what we have now at this stage is we start off with a big cell. It's got nice and big, so it's got lots of membrane to split in half. And it's got its DNA sitting there, and a copy of it's been made. Does that make sense? Okay, so what we get at this stage is a cell, and it's got a nucleus. And by the end of at the beginning of this stage, it's kind of loosely packed. And what's going to happen is, over the course of this stage, which we call prophase, because it's the first stage, what happens to the nucleus? The nucleus is going to, excuse me, the nucleus is going to dissolve. Why does the nucleus have to dissolve? So the DNA can be moved, right? And at the same time, the DNA is going to form into the chromosomes again. They're going to tightly pack. And there we are. So the nucleus dissolves. The chromosomes form. The centrioles migrate to opposite ends of the cell. We haven't talked about those yet. We have these two structures called centrioles. They're going to move to opposite ends of the cell. And they're going to start to produce something called spindle fibers. Sometimes you'll see the term astral rays. Kind of means the same thing. But you start to see this kind of thing, like that. And at the end of prophase, this is what it kind of looks like. This has started to happen. Why is this happening? Well, the goal is, what do we want to do? We want to take the DNA, we want to repack it back up so it's nice and tightly packed, and we want to take one copy of each chromosome and move it where? To opposite ends. Because what we eventually want to do is split the cell. And when we split the cell, we want to make sure that one copy of this goes down here and the other copy goes there. One copy of this is going to go down here, one copy is going to go there. What are the spindle fibers going to do as they get up here? They're going to grab on to chromosomes. And they're going to eventually shorten again and take the chromosomes and pull them apart. Does that make sense? So the first step is to tightly pack the DNA to keep it safe and make it easy to move. Get rid of the nucleus so it can move, and then start to grow the parts that are going to actually move them. That's pretty logical, right? And that's prophase. The next phase is called metaphase. And in metaphase, the chromosomes actually get connected to those spindle fibers. So let's draw that. What's going to happen here is, by the end of this, the chromosomes are going to be lined up at the center of the cell, what we call the equator of the cell, but basically means pretty much in the middle. And what did we call these pieces again? The centrioles. And then, this is kind of miserable, and I'm sorry, but holding the two copies of each DNA together is a structure called the centromere. And then we've got these spindle fibers that are going to connect. And of course, there's actually more than just two chromosomes, but they actually connect and grab onto the chromosomes, and they form out of both ones. Is it intimidating having such a good artist for a teacher? Do you feel like, oh my God, why would I even bother? I don't know, I miss her. What'd you say? I should be the art. Yes, yeah, look at me, oh my God, it's... You'd pay me. Okay, and I want to zoom in here and just kind of focus on this. What we end up with is the chromosome looking like that. Do you think Middleton's not here because she saw what a great artist I am and she's just too intimidated? See how quickly somebody just said, no, I do not think that. I do not think whatever's happening, it's not that. This is called the centromere. And this is a real problem in science where we just give things two names that sound like the exact same. But the spindle fibers connect to the centromere from each pole. And within the chromosome, remember, we have copy 1 and we have copy 2. Okay, so in prophase, we get the DNA ready to be separated. We compact it back into its chromosomes, we get rid of the nucleus, the centrioles move to where they need to be, and they start to make these fibers that can move out. In metaphase, the spindle fibers have made contact, and they line the chromosomes up at the center of the cell. There's no nucleus to be seen anymore, and the spindle fibers that connect to the centrioles grab onto the centromere of each chromosome. So they connect the middle of each chromosome to the two ends of the cell. And now we're going to have a tug of war. You can imagine, let's say that Trush and I had a tug of war with Matthew in the middle. If we pulled hard enough, could we both end up with an arm and a leg?
Yes.
Sure. Yeah, just basically, you can imagine it being like a tug of war over a person, and when you're done, the person is just cut clean down the middle. Everyone gets one of everything. It's kind of a dark image, and I regret saying it, but we'll move on. So, in anaphase, we actually get the centromere to split apart, and from the two, what are these called again? Centrioles. The spindle fibers shorten, and they pull their half of the chromosome in toward them, and the same thing happens over here. Okay, what do we have to do now? We're getting real close. Now we want to turn this into two cells. Connect these. We're gonna kind of connect them. We're gonna make sure they're together. We're gonna make sure they don't get away from each other. How do we like to hold the DNA to keep it safe and so it doesn't have to be tightly packed to stay safe? Yeah, what do we put the DNA in normally in the cell? In the nucleus. Right now we have DNA on both sides of the cell, but we don't have a nucleus. So we're gonna make a nucleus for each side. Does that make sense? And then we have one whole cell, but we want to have how many? Two. So we're going to make a new nucleus for each side. And then to make two new cells, again, knowing how the phospholipid membrane works, if you just squeeze the cell in the middle and can get it so that this side touches that side right down in the middle, you can simply split one cell into two. Does that make sense? So that's what happens during the finals phase which is called telophase or telophase. I never know how to pronounce it. Where around each of these, the spindle fibers break down and we get new nuclei. So there's one nucleus. And there's another nucleus. And then it will start to close in on itself like that. Matthew, are you pre-chuckling? Matthew, what do we call this? Go ahead. This is the cleavage furrow. Okay, what does cleavage mean? And I don't mean in the pejorative sense. What's a cleaver, for example? Yeah, which is used to take one piece of meat and turn it into two. It's cutting something in half. So cleavage just means cutting something in half. Right? Well, especially with what the diagram kind of looks like. Yeah, no, it's fair enough, right? I think scientists just have, dirty senses of humor. That's fine. But there we go. By the way, do you know cleave is one of my favorite words in the English language? And I word this carefully because I'm not going to say cleavage is one of my favorite words in the English language because that would make me sound creepy and inappropriate. But rather, cleave is one of my favorite words in the English language because it has a distinct character that only English can get away with. English is such a stupid, crazy, dumb language. Do you know that cleave has two meanings that are not just different, but completely actually opposite? If you look up cleavage in the OED, cleave or cleavage, but again, for this version, because I'm saying how much I like it, we're going to stick with cleave. If you look up cleave, one of the definitions is to break two things apart. Do you know what the next definition is? To join things together. It means its own opposite. It's an autoantonym. Autoantonyms are words that mean the opposite of themselves. Cleave, break apart, or put together. Literally the exact opposite. It's the same word. Literally, cleave can mean to stick to, or cleave can mean to pull away from. Can anyone think of another example of a word that means its exact opposite? Oh, you're breaking down over there, Katie. That's good. See if you can think of one. That's my homework challenge for you. Can you think of a word that means two things and those two meanings are opposite? I have a few in mind. Oh, what do you got? Yeah, you know what? I'll take it. I think that's a good example, actually. I like that one. So see if you can think of some other words. Autoantonyms. Cleave. All right. And then finally, we get, after mitosis, something called cytokinesis, where the two cells split fully apart and move away from each other. Cytokinesis. Cyto means cell. Kinesis means movement. It literally just means the two cells move away from each other. Oh, I just wrote cyto again. That doesn't help. Cyto means cell, and kinetics is motion. So moving. And so this is where the two cells simply split apart, and now we have two cells. And of course, once those two cells have moved apart, the DNA is going to go back into its chromatin form to just kind of do its thing. And now we have two daughter cells, and they are clones of the parent cell. What do we mean by clones? Yeah. Nobody in here is an identical twin, are they? Identical twins are clones of one another. Are you? No. Your dad is. And do he and his brother, like, how old's your dad now? Like my age or whatever, roughly like 40? Yeah. Do he and his brother look a lot alike? Because some identical twins, it seems like all through life they look very the same, and then some seem to... That's really funny. I went to teachers college with two girls who were identical twins, and they got one job and they just shared it between them. Isn't that amazing? So they would just like, if one of them felt like working that night, she'd go in, and if the other one wanted it, right? But they got a job, but like under one of their names, and they would just. I don't know if they did, but it's a good idea. Like one of you could study for biology, one of you could study for chemistry, do different seminar groups. Not a bad idea. You know, they probably did do that, but they didn't go around telling everybody. Anyway. Okay. I kept a couple of these because last year I had my grade 10s make little models of this, and these are actually really good. I think these are a nice little thing, so you can kind of see those here. We keep those here. I want you to be really comfortable with this as a concept because we're going to talk about this a lot moving forward, and we're going to use it to talk about more complicated things. Does that make sense? So you should know the stages. I should be able to say, hey, like what happens during metaphase of mitosis, and you should have a sense of like, oh, at this page, the chromosomes have all lined up, and they're ready to be pulled apart, but that hasn't happened yet. And then by anaphase, they've been pulled apart. And by telophase, it actually starts at the cleavage furrow to separate into two cells. And then finally, by cytokinesis, they actually separate into two cells. Questions about any of that? Comments on how beautiful my artwork is? Sorry? Oh, good. I'll look forward to that.
Thousands of clones working together to form one unit.
Yeah, that's exactly what happens. Now, we'll talk when we get to animal systems. Your body has a few things where it takes clones and then, well, does different things with them. Imagine if you had identical twins, but one of them decided to dedicate their life to being a bodybuilder, another one dedicated their life to being a professional eater, and the third, like one of these food competition people, and then the third one dedicated their life to, I don't know, to being a vegetarian and reading poetry. They'd look pretty different by the time they were done, right? And that's what your body cells do. Right? They're all clones of each other, but they dedicate themselves to different purposes. We'll talk about that.
Where did the third one come from?
Triplets. You can have more than two twins, right? What's the record for identical? Is it like... But is that identical? I never know what these things. Like when you get the big like quintuplets and septuplets and stuff, are they identical twins or